Hello students, again I am back with one of my topics in science in my channel, the report card. Now every day I have been posting the videos on topics of chemistry, okay, and I completed a playlist for acids, bases and salts, and now I have come back with another set of video, and it is an extension of the chapter Heredity and Evolution. In fact, when I started this channel with one of my friends, I started with the topic Heredity and Evolution in biology. Okay, but due to some certain unavoidable circumstances, I could not complete that series, and I completed till monohybrid cross. So those videos are available, and I will be making a playlist of all the videos on this particular chapter. And today I am moving ahead with that particular series only, and now today I am going to talk about dihybrid cross. Okay, so in my previous videos I told you about genetics, I told you about variations, I told you about alleles, I told you about the homozygous and heterozygous condition, the phenotype, the genotype, what are dominant traits, what are recessive traits, and what is the law of segregation or the law of purity of gametes. And definitely I told these all are Mendelian laws. So we were actually studying Mendelian laws, and like I told you about the monohybrid cross. Okay, so it is a cross or a crossing done between two individuals or two plants, observing the inheritance of one character. Okay, observing the inheritance of the contrasting traits of one particular character. So in the monohybrid cross, in the Mendelian experiment, the character that was taken was height of the plant. Okay, so we had tall plants and we had dwarf plants, and in the first generation we got all tall plants. We, in the sense that Mendel got all tall plants, and thus the law of dominance came into existence, that definitely the first filial generation had the genes or alleles of tallness and dwarfness, but the allele of tallness expressed itself, and thus it is known as a dominant allele. So everywhere, whenever there is a heterozygous condition, one of the trait is always expressed and other is not expressed, and that is the trait that is expressed is known as dominant trait. Okay, then besides that, there was another law, that is law of segregation. So what happened when Mendel continued this experiment, and when he crossed the plants of F1 generation, so both were hybrid tall. Okay, so the hybrid tall plants, when they were crossed amongst themselves, or there was a self crossing or self pollination was initiated between them, then in that case what happened, the plants of the F2 generation or the second filial generation, these plants were tall and short, both. Like Mendel got both the type of plants, right? So the ratio was 3:1. Okay, now Mendel was basically surprised on getting the dwarf plants, because initially when Mendel studied the first generation, he thought that all the alleles of dwarfness have either mixed with the alleles of tallness, or maybe they must have perished or something. But when he got dwarf plants, that was 25%, then he came to a conclusion that the alleles of dwarfness still retained their identity. Okay, so even when they were present with the allele of tallness, they maintained their identity. And how could they express themselves? They got segregated during the gamete formation. Okay, so this is what happens during gamete formation, because gametes are haploid. Okay, so a diploid cell which has homologous chromosomes having alleles of the contrasting traits which are present in both the chromosomes at the same locant, then during gamete formation, those homologous chromosomes, they separate and they go into each gamete. So this way the gametes basically get only one allele. of each trait or in fact of each character, right? So this is called as segregation. So the alleles always get segregated during gamete formation, right? So this was basically the monohybrid cross. Then Mendel thought, what if we are taking in more than one character? Okay, so he actually took two characters into consideration, and whenever a crossing is done between two plants or two individuals to observe the inheritance of two characters, okay, or you can say to observe the inheritance of contrasting traits of two characters, we call it as dihybrid cross. Okay, so in a dihybrid cross which Mendel basically performed, he took two characters into consideration. Now what were these two characters? One was color of seed, okay, color of seed, and other was the shape of seed. Okay, these were two characters which were considered. Now when we talk about the color of seed, Mendel had two colors. There were two colors of seeds which were found. One was yellow and other was green. Okay, so there are two colors of seeds that are found in pea seeds or pea plant rather. Okay, then when we are talking about the shape, again there are two types of shapes. One is round and other is wrinkled. Okay, wrinkled. So these were the four traits that Mendel got, two of color and two of shape. Okay. Now what Mendel did was, Mendel took seeds which had yellow and wrinkled. Okay, so when the color was considered, it was yellow and the shape was wrinkled, and he crossed it with seeds which were green and round. Okay, which were green and round. Okay, and again, as if you watch my previous videos, you will find that these which we call it as P1 generation or parental generation, the traits are always pure. Okay, the traits are homozygous and pure traits, right? So yellow and wrinkled, green and round, these were the two traits that were taken. And then in F1 generation, what did Mendel find? Mendel found that all the seeds had yellow color and round shape. Okay, yellow color and round shape. So all the seeds which Mendel got in the first filial generation had yellow color and round shape. Okay, so this established the fact that law of dominance prevails, at every step. And so whatever type of cross it is, if it is a monohybrid or a dihybrid or a trihybrid, polyhybrid crosses also, everywhere the law of dominance prevails, right? So whenever there is a heterozygous condition, the dominant allele will always express itself, right? So here the dominant allele is yellow, and as far as the color is concerned, the dominant allele is yellow, and as far as the shape is concerned, the dominant allele is round. Okay, so yellow and round, these were the seeds that Mendel obtained, right? So now let us just do the genotypic crossing for this particular for this particular dihybrid cross. Now please watch my previous videos where I have told you how to write the genotype and everything. That's very important. So over here I am not going to repeat all these things, okay, because repeating everything will unnecessarily extend the length of the video and the time. So please watch my previous set of videos on heredity and evolution. So, like if I am going to write yellow and wrinkled, okay. So first of all, I want to write the genotype for yellow. Okay, so when I am writing the genotype for yellow, how am I going to write? Now yellow is a dominant trait, and I told you that we have to take the first alphabet of the dominant trait, right? So the genotype for yellow will be written as capital Y, because there are always two alleles which exist together for a particular character. Okay. Similarly, when I have to write green, so how am I going to write green? So I told you that the recessive trait of the same character will be represented by the same alphabet. So green is again a color. Okay, so we are going to represent it with the same alphabet, but yes, it will be a lowercase because it is a recessive trait. Okay, so this denotes green color and this denotes yellow color. Then after this we have the shape. Now the shape round is the dominant trait. Okay, so we will write round as capital R. So over here we are not the Mendel actually took yellow and green seeds. Yellow and wrinkled seeds. So I am going to write round over here as capital R. Okay, so this represents round. So this is green and this is round. Okay, green and round seeds. Okay, but then the recessive trait is wrinkled, so wrinkled will be represented by small r. So here we have yellow and wrinkled seeds. Okay, so is it clear to everyone? So how to write the genotype? I have already explained it in my previous videos, so please watch those videos so that you are aware of how to express. And I have in fact told you that this is a way which is actually prescribed for CBSE. But when I am teaching in IGCSE or Cambridge or AS level and A level, there is another way of expressing the genotype. Okay, so that is another way where we write the particular genotype in the form of a superscript. Okay, so that's another way. So this is the way prescribed in CBSE, right? Okay. Now this yellow and round seeds, and this is yellow and wrinkled and green and round seeds, right? And this is the P1 generation, parental generation. Now the second step, as I told you, that whenever we have to do crossing, we have to first make the gametes. Okay, because the crossing is always between the gametes, right? So how are we going to make gamete? Now in each gamete we will have the alleles of all the traits, isn't it so? So if there are 10 traits, the alleles of 10 if there are 10 characters rather, so the alleles of 10 characters will enter into the gamete, right? Even though it is one allele. So here we can say 2 characters are there. So alleles of both the characters will enter. So one allele of color and one allele of shape. Okay, similarly we will have another one allele of color and one allele of shape. So we can have 2 gametes, right? So over here this, and then again this and this, that is capital Y and small r. Fine. Now similarly here also it is small y and capital R, that means green and round, and here also small y and capital R, that is green and round, right? So these are what? These are the gametes. Okay, you have to write down the gametes because the crossing is done between the gametes. Okay, so now let us just do the crossing. So either you can make a Punnett square to do the crossing, you know what to do in the Punnett square, but here I am not making the Punnett square, or wait, let's make it, because in this dihybrid cross it's better that you make a Punnett square and show the crossing. And again I don't have to explain the Punnett square because I have already explained it in my previous videos. Okay, so please I don't want to extend this video unnecessarily, so that's why I am not explaining the things that I have already explained in my previous videos. So see, this is it. On one side, suppose if we take it as female and we take it as the male. Okay, so on one side I have written the gametes of the female, and the other side I have written the gametes of the male, and then let us cross these gametes. So capital Y, small y, capital R, small r. So why? Because for one particular character, both the alleles have to be written together. It's homologous, है ना? So whenever there is homologous chromosome, the alleles of the same character, they lie side by side in both the chromosomes at the same location, right? So that's why we are writing it together. Okay, so capital Y, capital R and small r, and similarly the same way we are going to write here. Okay, like this. So this is again this is our F1 generation in dihybrid cross, and we see all the plants that we have received, they all are what? They are hybrid yellow and round. Okay, they have hybrid yellow and round seeds. So just now I am winding up over here. In my next video I will be telling you about the F2 generation. Okay, the dihybrid cross, the extension of the same. Till then, take care.